4th Annual James R. Killian Jr. Faculty Achievement Award Lecture. I'm Krishna Rajagopal, Chair of the Faculty. Before we begin, just to tell you the program, it's my guess that the lecture and the Q&A afterwards will go till about 5:10, 5:15, along those lines. And then afterwards, there will be a reception downstairs in the Bush Room to which everyone is invited. The James R. Killian Jr. Faculty Achievement Award was established in the spring of 1971 as a permanent tribute to Dr. James Killian, who was president of MIT from 1948 to 59 and chairman of the corporation from then until 1971. The purpose of the award is to recognize extraordinary professional achievement by MIT faculty members and to communicate their accomplishments to members of the Institute writ large. The title of Killian Lecturer is the highest honor that the MIT faculty may bestow on one of its own. It is our privilege today to honor and welcome Tyler Jacks, the David H. Koch Professor of Biology and the Director of the Koch Institute for Integrative Cancer Research at MIT. As noted by his colleagues on the Killian Award Committee, Professor Jacks has long been a pioneer in the MIT cancer research community. His early achievements in genetically engineering mouse models that closely replicate the human disease allowed certain types of tumors to be studied with greater reliability. And today, the models he developed are used in labs worldwide, contributing to a better understanding of disease pathogenesis and progression and enabling the testing of novel anti-cancer strategies. Both in and out of the lab, Professor Jacks is described by colleagues as bold and visionary. His nominators point out that it takes a village of passionate and dedicated people to invent solutions for the many cancers that afflict our society. And they point out this is exactly what he has created over in Building 76. Yet, as one colleague commented, successes like the Koch Institute do not just happen. They are created through dynamic leadership. And this is one of the qualities for which we honor and thank Professor Jacks today. Since 2001, when he was selected to direct the center that would become the Koch Institute, he has fostered transformative changes by cultivating multidisciplinary collaboration between cancer biologists and engineers. Today, the Koch Institute houses dozens of faculty research groups and serves as an organizing body for more than 500 researchers at MIT. Faculty and students cite his mentorship and his commitment to bringing the different cultures and languages and research agendas of these two communities, these two worlds together. He also does a marvelous job of reaching out to the larger world outside MIT, communicating challenges overcome and lessons learned at the cancer research frontier to broad audiences. For example, and I can personally recommend this, through a TEDx Cambridge talk that he gave back in September of 2014. Professor Jacks joined the MIT faculty in 1992, and in the years since, he's been recognized with numerous awards. He's been named a Howard Hughes Investigator and received the Paul Marks Prize for Cancer Research. He was elected to two of the nation's most prestigious academies, the National Academy of Sciences and the Institute of Medicine, in 2009. That same year, he was elected president of the American Association of Cancer Research, and he was appointed chair of the National Cancer Institute's National Cancer Advisory Board in 2011, and in 2012 was elected to the American Academy of Arts and Sciences. So it's a privilege and a pleasure to welcome Tyler Jacks to the lectern.
Thank you so much.
And now I get to actually give you the certificate.
Okay.
I have the mic for another second.
Okay.
So, and Tyler, it is my privilege to represent the MIT faculty in honoring you with the James R. Killian Faculty Achievement Award, through which we recognize your influence on the field of cancer research achieved through your own pioneering work and your visionary leadership to unite MIT's cancer biologists and engineers in a collaborative, multidisciplinary research community. Congratulations.
Well, Thanks, Krishna, for that very, very nice and generous introduction. At the appropriate time, I'm happy for you to deliver my eulogy. I want to thank Professor Rigobon and the Killian Award Committee for selecting me for this honor. It's a tremendous honor, I must say, in part because it's given to me by my peers, and in part because it represents the great tradition of our university. I'm frankly blown away to see my name on the list of Killian Award recipients. I'm not sure I belong there, but I'm happy to be there just the same. This award is particularly meaningful to me because I have extremely long history at MIT. I came here in 1988 to pursue my postdoctoral studies with Bob Weinberg in the Whitehead Institute and joined the faculty in 1992. So that's a span in total of 28 years, quite remarkable. Of course, I started when I was 9, so that's okay. My brother is a graduate of this institute, class of 1976. And some of you know that my father was a faculty member here for 30 years, starting in the late 1950s until his retirement in the 1980s. Rumor has it that I was conceived on the MIT campus. I'm not sure that's true. It's a story for another time. But my dad used to bring me to MIT when I was a kid, and I can tell you that Kendall Square in the 1960s was a very different place. There was literally one place to eat. It was the F&T Diner. Some of the old-timers will remember the F&T Diner. It's where the tea station is, and my dad and I would go there each time, and we would always have pastrami sandwiches, and I can still remember the taste of those pastrami sandwiches today. And my education at MIT, from MIT, started very early as well. I learned to play poker from my father's colleagues around the table at our house at their monthly poker game, and that included people like Frank Fisher and Bill Pounds and Peter Gill, and Howard Johnson actually used to come. Howard Johnson, former president of MIT. That was in a simpler time, Raphael, when the president could gamble with the members of the faculty. My dad passed away about 15 years ago, but I'm quite sure he would be extremely proud of today's events. I want to thank you all for coming. I know that you expected a lecture on gravitational waves, but I'm glad you're here anyway. I want to thank the Koch Institute folks who gave me a rousing send-off from the building. Much to my surprise, I walked out the door. There were 200 people in a gauntlet lining the courtyard as I walked my way over here. So thanks for that. That was really quite a send-off. I want to thank David Koch for making the time to come up from New York to be with us today. I want to give a special welcome to my wife, Laura, and my daughter, Livy, and thanks to them for their never-ending love and support. And I want to make a special thanks to Joe Cohen. Where is Joe? I hope he's here. Joe Cohen was responsible for all the logistics related to today's event, including the advertisement, and Joe advertised the hell out of this thing. I couldn't turn a corner on campus without seeing my face. I know what it's like now to be one of America's most wanted. So thank you for all of those things. And certainly thank you for the recognition. I'm being recognized for two things, I guess. They were mentioned in Krishna's introduction. One is the science that we've performed, and the other is the Koch Institute development. And I'm going to talk a bit about both of those things today. With respect to the science, I think it's important to point out that scientific achievement is not a singular activity. It's the collective activity of many people. And I've been extremely lucky to attract outstanding people to work for me over the last quarter century. Graduate students like those shown here, the names and faces of the past and present graduate students from my group are shown. We get wonderful students at MIT, undergraduates and graduate students, and they contribute mightily to the work that we perform in our laboratories. Likewise, I've had the privilege of training a very large number of outstanding postdoctoral fellows. Again, the names and faces of the individuals presently in the lab and previously in the lab are shown here. And these folks, the postdocs and the graduate students, have continued to do great research in their independent training and independent careers, contributing to discoveries in cancer research around the world, across the country, really carrying things forward. And I would say one of my proudest, if not proudest, achievements as a faculty member is being able to train these wonderful people. This is the greatest product of our laboratory's research. Students and the postdocs often get the headlines, but importantly, they are assisted by technical help, technical assistance. We've worked with a number of great technicians over the years. Their names and faces are shown here. They deserve a lot of the credit as well. And over the years, we've had wonderful undergraduate students and summer students, high school students work with us. And they contribute as well, both in the science that they pursue and also as mentees for the mentors in the lab so that they can learn how to become better teachers and to learn the skills of mentorship. And finally, the lab runs because of great people who help administer the lab, and I've been assisted by wonderful people over the years, shown here, my administrative assistants and lab managers. I want to make special reference to Judy Teixeira, who's sitting in the second row here. Judy's been with me for 15 years as my administrative assistant. The smirk on her face sort of suggests that she's tired of hearing yet one more thing that I need her to do. But thank you, Judy, for all that you've done over the years. Now, the introduction referenced the development of the Koch Institute, and the development of the Koch Institute likewise was a shared experience. It was a joint effort involving many, many people, including, very importantly, Jackie Lees, who's the associate director of the Koch Institute and was the associate director of the Center for Cancer Research starting back in 2001. And Jackie and I really conceived of changing the course of cancer research and including the engineering community in that new direction. We were joined at that time, 2007, by Dane Wittrup, who likewise made major contributions to the directions that we finally took and the planning that we made at that time. Phil Sharp was a major force in that. Bob Langer, Doug Laufenberger, and many others on our faculty contributed significantly to the progress and planning that we made. Cindy Quentz is the Assistant Director for Administration of the Koch and does a wonderful job overseeing the headquarters staff and the operations. Anna DeConnick and before her Robert Urban have served as executive directors of the Koch, taking care of many of the outstanding outreach efforts that we make in various directions. So I'm very indebted to all of them and many others who've made the Koch Institute what it is today. This wouldn't have happened if not for the support and encouragement and massive effort by the MIT administration. This happened in 2007 while Susan Hockfield was president, Raphael Reif was provost. And they, too, contributed to the idea of bringing scientists and engineers together under one roof. If not for them, we wouldn't have done it. And they continue to be tremendous supporters. Susan, whose office is now in the Koch Institute, Raphael, as president, has remained a steadfast supporter of the Koch Institute. So I'm very grateful to the MIT administration for helping us do this. I'm grateful to the deans that I've worked with over the years in science and engineering, vice presidents for research, Claude Canizares, Maria Zuber, who've been tremendously supportive. And we wouldn't be here if not for one specific individual, and that's David Koch. David was remarkably generous in providing the funds necessary for us to build the building that we now occupy, to provide funds to equip the building that we now use, and has also funded 6 professorships along the way. So David has been remarkably generous to us here at MIT for cancer research and frankly around the country in support of cancer research. So thank you to you, David. The idea of doing cancer research in the way that we now do it in the Koch Institute takes advantage of great strengths that we have, somewhat uniquely here at MIT. Cancer scientists working together with cancer-oriented engineers to encourage new approaches to long-standing problems in cancer. The idea was that there would be opportunities at the interfaces of various disciplines in basic science and engineering to approach problems in cancer that had been very difficult using singular approaches in the past, that there was some magic at this interface, and that we could therefore make new discoveries, different kinds of discoveries, and new inventions and new technologies through a very deliberate effort in collaboration, working across disciplines to take advantage of this rather unique mix of individuals that we have in the Koch. And moreover, that we could mobilize our efforts beyond those discoveries and new technologies and deliver those to the benefit of patients. Not to stop at the discoveries at the bench, but actually to translate those discoveries to the benefit of cancer patients, to change the lives of patients, to change the course of cancer, And frankly, I'm pleased to say that we're starting to do that, and I'm extremely proud of that as well. So 2007, we started thinking about how to develop the Koch Institute. We planned it over the next couple of years, again, with contributions from many people, including folks in the planning office here at MIT. And finally, in late 2010, early 2011, we opened the doors to the new Koch Institute building. We brought this project in, David's proud to say, under budget and ahead of schedule, quite remarkable. And it was celebrated, as you can see here on the front page of the Boston Globe, above the fold, a new style of cancer research, coming together, bringing individuals from diverse backgrounds together to approach the problems in a new way. I'd like to point out that in this same page that this doctor who was indicted had nothing to do with us. Quite sure that he was a Harvard doctor. So we moved in, and this is what things look like in Building 76 now. It's a beautiful building. You've all seen it. Some of you work inside of it. And around the rim are the faculty who occupy this building. In blue writing are the science faculty, and in green writing the engineering faculty. We blurred the lines between these disciplines now, so it really isn't appropriate to use different shading for these folks. But this is a remarkable collection of people, extremely talented in their fields, and also quite distinctive in their willingness to broaden and engage researchers across many different areas of science and engineering and technology development. They're very ambitious, they're very creative, they're a bit unconventional. Frankly speaking, you don't find cancer research institutions like this anywhere else. Cancer research institutions in most places look like a swim meet, where everybody's more or less doing the same stroke, staying in their lane with their heads down. Cancer research at MIT and the Koch Institute looks quite different. Nobody's doing the same stroke. Some people are doing strokes you cannot recognize. People are ripping up the lane markers. Angie Belcher is lighting them on fire. Mike Yaffe and Paula Hammond are diving off the high board doing cannonballs into the water. Cancer research in the Koch Institute looks different. It's frankly a frothy mess. And we like it that way. We're doing different things, and we're changing the paradigm. We're changing how we're approaching the cancer problem, bringing the inventiveness, the innovation, the spirit and creativity of MIT together to solve these long-standing problems in a different way. Now, it's not only about what's happening inside the building. The Koch serves as the National Cancer Institute designated cancer center for MIT, and therefore we organize the cancer research efforts on campus. And in this way, we reach out to cancer researchers across the campus. And there's more than 1,000 cancer researchers at MIT, in fact. A full 27 faculty are extramural members of the Koch Institute. It's very much about collaboration across disciplines, across buildings, across the campus. And collaboration is very important to what we do. It allows us to take advantage of the creativity of our colleagues independent of where they come from. And there is this magic at the interface, if you're open to seeing it, this collective creativity. We see it at play every day. And don't discount the importance of beer. Extremely important as well. The collaborations include the folks within our communities, but importantly, and especially as we translate our ideas out from MIT beyond, we actually need to collaborate with other people. We don't have a cancer hospital at MIT. We don't take care of cancer patients here. And so in order to be able to do more clinically relevant research, we must reach out. And we've done that in lots of different ways, in particular with respect to the Bridge Project, which is a project that we have in collaboration with the Dana-Farber Harvard Cancer Center. And here we've funded teams of investigators. Soon that number will be up to about 25 teams of investigators between MIT and Harvard tackling more clinically proximal problems. And this, again, brings our ideas and our discoveries directly to the benefit of patients. We also reach out to industry, and these interactions are also really important. There's only so many things we can do in our labs. Sometimes we need the capital and the expertise of people from industry. And so we've engaged with biopharmaceutical partners in a fairly significant way. And we've also spun out a bunch of companies. This is a list of companies that have spun out of the Koch Institute, intramural and extramural labs, only since 2007. There's about 42 companies shown here. Amazing to me. Bob Langer has started 35 of them. But We're very proud of this fact. Again, it advances our work more quickly to where it can make the biggest difference. So these kinds of approaches, organizational structures are really important, but so too is the culture. Phil Sharp reminded us early on that culture beats strategy every time, and he's absolutely right. And so we've tried to build a culture of collaboration within the Koch. We have regular meetings of students and postdocs presenting their ideas to the audience at large, an audience that involves people from different disciplines, and each time they learn something new, and each time they walk away thinking about something differently. This is part of the success. And they also teach each other in the process. These are two examples of programs run by students and postdocs in the labs. Again, teaching each other about the problems they know and the others might not know so well. And so this kind of environment, this culture that we've created, I think is part of the reason why the Koch has worked. And I'm extremely proud and grateful to all the people who've contributed. All right, so that's the Koch introduction. We're now going to switch gears and talk about science. And because we're going to switch gears and it's at MIT, it's appropriate that we start with a test question. So here we are. Which of the following diseases claimed the most lives in 2015? Is it A, HIV/AIDS, B, tuberculosis, C, malaria, or D, cancer? Who thinks A? B? C? D? Livy, what's the answer?
D, cancer.
Of course it's cancer. It's a cancer lecture after all, for God's sake. Cancer claims more lives in the world than the other three combined. You might find that hard to believe, but about 8.2 million people lost their lives worldwide to cancer. That number is actually going up, sadly. So cancer is a big problem. It's the kind of problem that MIT should be investing its time and energy and resources to solve. So I need to give you a little bit of a primer. This is an audience that is diverse, coming from different areas, so I want to start with the basics, and then we'll move ahead to talk about some of the cancer research that we pursued in our lab. So cancer cells arrive from normal cells and acquire changes that allow them to divide inappropriately. That's what cancer is. It's an inappropriate accumulation of cells. Too many cells in the blood, too many cells in a tissue, a mass. This process of uncontrolled cell division and cell accumulation is driven by changes in genes, changes in the genes that control the normal behavior of cells. And there are two classes of genes that are classically involved in cancer development. Oncogenes, which drive the process forward. These genes become activated, hyperactivated in cancer. And then there are tumor suppressor genes that normally act to restrain this process. They get inactivated in cancer. They get eliminated. So these two classes of genes underlie the development of all cancers. This is the genetic basis of the disease. In addition, cancer cells don't grow up in isolation. They grow up inside tissues in your body. And as such, they interact with the, quote, normal cells of your body too. And these interactions turn out to be extremely important. They help the incipient cancer cell evolve. And in some cases, they try to inhibit the development of those cells. Immune cells, for example, can both promote and try to eliminate cancer cells during their evolution. So cancer is a complex process. The inner workings of the cancer cell, as well as the interactions between the cancer cell and the cells that surround it. In addition, cancer cells typically don't stay where they start. They move in the process of metastasis to other sites in the body. And metastasis is actually the lethal phase of cancer. About 90% of cancer deaths are attributable to the metastatic phase of the disease. It's a phase that we don't actually understand very well, and we at MIT are actually devoting a lot of energy to understand that process better. I'm going to return to this slide at several points in the lecture to show you how we in the lab are approaching these questions through the models that we've developed. A bit more background. These genes that I mentioned lie within your chromosomes. When you're born, your cells have the proper genetic dowry assembled on 23 pairs of chromosomes. Those chromosomes are painted different colors here. This is what a normal karyotype looks like. Cancer cells have problems with their DNA. Here's an example of a cancer cell which has way too many chromosomes. You can appreciate that. This is called aneuploidy, Angelica Ammon Institute is studying the details of aneuploidy and how it contributes to cancer. You can also see certain structural changes in the DNA, like this chromosome here, which is made up of two different colors. This is what's called a translocation, where one piece of one chromosome has been stuck to a different one. This creates a new gene, likely an oncogene, which is driving these cancer cells forward. Here's another example of a chromosomal spread of a cancer cell, and you can see that these chromosomes are all screwed up. There's multiple colors. This is genetic haywire. It's amazing that cancer cells can actually live with all this chromosomal damage, and yet they do. They grow and ultimately cause severe damage. Now, within all of those changes, we now know from massive sequencing efforts of the genomes of thousands of cancer specimens, much of that done at the Broad Institute, that there are about 600 genes that are mutated at significant frequency across all human cancers. So 600 of the about 22,000 genes that are in your genome are directly contributing to one or another form of cancer. That's a pretty large number. And that actually represents some of the difficulty, some of the challenge that we cancer researchers face, is to figure out the contributions of all of those different genes in order to be able to understand the process and hopefully do something about it. One of the promises of figuring that out, once you understand the molecular basis of cancer, is you might be able to treat it better. If you can figure out exactly what's driving a cancer, you might be able to make a medicine that overcomes that effect, that directly inhibits that effect. This is the so-called idea of precision cancer medicine, and we're in the era of precision cancer medicine today. There are new drugs that can counteract the effects of particular changes in certain genes in certain cancers. Importantly, among the earliest of these precision cancer medicines, two were based on discoveries made at MIT. HER2, a gene discovered in Bob Weinberg's lab, is the target of a breast cancer drug called Herceptin. It works extremely well in women who have that particular alteration. It's useless for women who don't. BCR-ABL is a small molecule drug that inhibits the function of an enzyme that's overactive in certain leukemias. The enzyme ABL was discovered in David Baltimore's lab here at MIT. Crizotinib treats a certain form of lung cancer based on a particular alteration. I'm particularly proud of that one because the international study that was done to lead to the approval of this drug was carried out by Alice Shaw, who used to be a postdoc in my lab and is now an independent investigator at the Mass General Hospital. So they're in an era now where we've translated this basic understanding of cancer genetics to some, but not yet enough, new medicines that are more effective and less toxic than standard chemotherapy. Now, this process of cancer genes and cancer genetics has a long history. Our understanding goes back many, many years. It actually goes back to this institution. The first cancer gene was discovered by Bob Weinberg. He found it in a study published here with Sheiho Shi while Bob's lab was still in the cancer center. This was the first demonstration that cancer cells, human cancer cells, have altered genes. This really set the field in motion and frankly set my career in motion. I was an undergraduate when this study was published, and Bob came and gave a lecture in a course that I attended and described this work. And it was based on that work that I decided that I would become a cancer researcher. So I owe it all to you, Bob. A few years later, Bob's lab cloned the first tumor suppressor gene in collaboration with Ted Daraja's lab. This is the retinoblastoma tumor suppressor gene. A few years later, David Hausman's lab cloned the second or third tumor suppressor gene, William WT1. So MIT researchers have contributed significantly to the discovery of cancer genes over the years. The retinoblastoma gene falls as a good example of a gene that's mutated not only in sporadically occurring cancers, where there's no family history of the disease. This gene is responsible for the development of a rare tumor of the eye, retinoblastoma. But also you can inherit from one parent a defective copy of this gene and thereby have a familial predisposition to retinoblastoma. And that disease looks something like this. This is a child with so-called bilateral multifocal retinoblastoma. This child has tumors in both eyes, probably about a dozen in total. And this is due to the fact that the child has inherited one defective copy of the retinoblastoma tumor suppressor gene from one parent and is therefore just one mutational event away from lacking this gene altogether. And when that happens, the tumors develop. When this work was done in Bob's lab, I decided that this is what I wanted to study. And I decided actually to come back to Boston, come back to MIT, and join Bob's lab to study this disease, and particularly familial predisposition to cancer, the genetic basis of cancer. And I used techniques that had been developed by other people in the field to allow us to manipulate the genes of mice in particular. We can do this in other species now, but at the time we could really only do it in mice. I won't go through the details of these studies, but suffice it to say that we can manipulate the genomes of mice by adding extra genes, oncogenes, or deleting certain genes, tumor suppressor genes, and by virtue of that make those mice cancer prone. And that's what these two studies published by other labs demonstrate. So I decided that I would use the techniques of genetic engineering in mice to make models of cancer that we could then use to study the disease process in ways that are difficult to do in human beings. So the idea was to create genetically engineered mice with mutations in cancer genes, create models of various types of cancer, and then study those models. And over the years, we've studied many different cancer types. Lung cancer, which is going to be a theme of today's lecture. We also study pancreas cancer, sarcomas, ovarian cancer, brain cancer, retinoblastoma I mentioned, and several others. Now, once you have these strains of mice that are genetically engineered to be cancer prone, you can use them for various things, including, for example, studying tumor development. And that was really the main purpose at the beginning. We don't understand the process of tumor development well enough. It's hard to study that process in a human being. But with a mouse model, you can do it because you can access the disease at various stages. You can look at the disease cells in ways you can't easily do in people, and so forth. So you can understand what we call tumor progression through the analysis of these mouse models. In addition, once you have them, they can be very useful practically. You can use these models to understand early detection. Maybe I can find the disease at an earlier stage than it becomes clinically evident. That would be highly useful. We'll return to that topic. Maybe we can figure out new therapies to treat the disease more effectively. If I understand the process better by these mouse models, maybe I can use a new therapy to block it and then use that new therapy in humans more effectively. Or maybe I can prevent the disease altogether. If I understand the process really, really well and be able to anticipate it, maybe I can actually intervene before it gets to an advanced stage in the form of cancer prevention. And we'll return to those topics as well. So the first mouse I made, which by the way took 4 years of my life, back-breaking effort, Bob will remember, he's smiling. Thanks a lot for that. Nothing worked for me in Bob's lab. I blame him. Nothing worked for me. But we struggled on and we made this mouse that had a germline mutation in the retinoblastoma gene with the full expectation that the animals would develop retinoblastoma, tumor of the eye. In fact, they did develop cancer, pituitary tumors at 100% frequency, thyroid tumors at 50% frequency, but amazingly enough, not a single one of them developed retinoblastoma. And frankly speaking, that was frustrating. Now, we went on over the next several years to figure out why it was that mice with a germline mutation in retinoblastoma, the genetic analogs of that child that I showed you a few minutes ago, actually don't develop retinoblastoma. And the answer is because there are 2 other retinoblastoma-related genes that go by the imaginative names of P107 and P130. These are structurally similar to RB and functionally overlapping. Some of that work shown by Jackie Lees here at the Koch. And it turns out that in a mouse, for some reason, in the mouse, they can cover for loss of RB function. So it turns out that you have to get rid of 107 or 130 in combination with RB. And when you do that, finally, in the hands of David McPherson in the lab, you get retinoblastomas. It took 15 years for us to get there, but we eventually succeeded. And so by sort of understanding the underlying biology and the underlying genetics, we could improve these models over time. And that's been an ongoing theme for us. A major focus since I moved to the Cancer Center and then on to the Koch has been lung cancer. Lung cancer is a major cancer type in the US. It's responsible for about 160 cancer deaths in the US, more than 1 million worldwide. Of course, smoking is a major carcinogen for lung cancer, responsible for about 90% of cancer deaths. There are various types of lung cancer, small cell lung cancer, non-small cell lung cancer. You might have heard these terms. The type that we study most extensively is called adenocarcinoma. It's one of the non-small cell lung cancers. It happens to be the most frequent of the lung cancers. And we know a bit about the genetics that underlie this form of lung cancer. We know based on genome sequencing analysis, for example, that 2 genes are frequently mutated. KRAS, which is one of these oncogenes that drives the process forward, and P53, which is one of the tumor suppressor genes that that normally blocks the process and is inactivated in cancers. I also mentioned a couple of other genes here, EGFR and ALK. These occur at lower frequency in lung cancers. I mentioned them because they are the target of some of these targeted therapies for lung cancer. There are drugs that treat these hyperactive genes. But based on the frequent mutation of KRAS and P53, we decided that we would study those genes in the development of lung cancer, and we made a mouse that we study now very extensively in my lab. There's a little bit of scientific detail on this slide. It doesn't really matter. Suffice it to say that we made a mouse that has genetically engineered copies of the KRAS oncogene and the P53 tumor suppressor gene. But in the native state, those don't do anything to the cells of these animals, including the cells of the lung. However, they're triggered to be activated in the case of KRAS and inactivated in the case of P53. And the trigger occurs by virtue of these little triangular shapes here. These are called LOXP sites. And when this enzyme called CRE acts to loop out those sequences, this KRAS gene goes from off to on, and the P53 tumor suppressor gene goes from on to off. And that's enough to initiate a cancer. And therefore, over time in these animals, we get lung tumors in the lung. And over further time, we see these tumors progress to metastasis. And so this is a model that we've now studied extensively to understand the various steps of lung cancer development. We've studied this many ways by investigators, some of whom are in the room, over many years. This is the contributions of the KRAS oncogene to the early stages of tumor development. This is actually a baby picture of a tumor contributed by Carla Kim, sitting right over there. This is the very earliest steps of response to KRAS activation, hyperproliferation of those cells. They form these little layers of abnormally overgrowing cells, benign tumors called adenomas that progress to larger adenomas. And we now know that if you inactivate the P53 tumor suppressor gene, those tumors will progress from a benign state to a malignant state. They go from benign to real cancer. P53 controls that transition. And over the years, others have contributed our understanding of many of these various steps, these arrows. We're developing sort of a molecular cartography of cancer development here, filling in the pieces of this puzzle. And we now understand it actually quite well. Now, why do we bother to understand what these steps are, these various active pathways, genes that become hyperexpressed? And the reason is, as I mentioned before, that if we can understand that in detail, in the mouse model and ultimately in people, we might be able to treat this disease more effectively. Now, I work on mouse models, and many people believe, and I bet many of you realize, that there's a long history of slamming mouse models of cancer. Many people think that it's really easy to cure cancer in mice, and therefore it's not really worth doing the experiment in mice if it's not going to prove true in humans. In fact, those statements are based on primitive models, primitive mouse models of cancer. These models are much more faithful to the actual disease. They look much more similar, both genetically and histopathologically, and related to the various cells that are interacting with the tumor. And so for all those people who think that it's really easy to cure cancer in mice, I say, cure this. And I've actually put my money where my mouth is. I've offered $10,000 to anybody who can cure this model. I never offer my own money. Today I will offer President Rife's money. He's tapping David Koch on the shoulder, I'll have you know. Many have tried, no one has succeeded to date. It's very difficult to do. Cancer is a tough challenge. We've tried as well. Here's two quick examples. Inhibitors against one of those pathways called NF-kappa B, inhibitors against another of those pathways I mentioned that gets hyperactivated, Wnt. These extend lifespan of mice. As you can see here, animals that are untreated succumb to their disease, in this case within about 100 days. If you treat with this small molecule drug or this small molecule drug, the animals live a bit longer. This is clearly not curative, but it's a step in the right direction. And we think that these treatments, perhaps in combination with other treatments, might actually get us to something that could be useful. And we're encouraging our pharmaceutical partners to pursue these drugs, actually, for the study of treatment in humans. These two drugs fall into a class, we call them small molecule inhibitors. They're useful in binding to enzymes that get hyperactive in cancer cells and turning them off. They fit like a lock and a key. There's another class of cancer drugs, the antibody-based drugs. These are much larger biological molecules. Herceptin is an example. They can bind to proteins on the outside of cells that get hyperactivated and turn them off. These are good, and where they can be used can be powerful, but they're quite limited. It's not always easy to find an enzyme that's hyperactive in cancer cells in order to drug it, or to find a protein on the surface of cells that you can inhibit by an antibody. It would be nice if we had a tool that we could use to turn off the expression of all genes that are hyperactive in cancer cells. And it turns out, in part based on discoveries from MIT alums, including folks trained by Bob Horvitz and Phil Sharp, that there is a new class of drugs emerging, so-called RNA-based drugs, small RNA molecules that can bind to and inhibit target mRNAs and cause either their functional silencing or their inactivation, their elimination. And if you could get these small RNAs into cancer cells, you might be able to turn off an oncogene. There's another class of RNAs called microRNAs, which can silence pathways that get hyperactivated when tumor suppressor genes are lost. If you could put a microRNA inside a cancer cell that lacked a particular tumor suppressor gene, you might be able to correct the defect caused by the loss of that tumor suppressor gene. And so we decided some years ago to see if this could work, in particular using these small interfering RNAs to target RAS in this model, or a microRNA that can substitute, at least in part, for the loss of p53. Now, the difficulty is that it's hard to get these microRNAs into cancer cells in the body. If you stick a naked RNA into the blood, it gets degraded. If you package it up in certain things, it mostly goes to the liver. It's fine for treating liver disease, and Phil actually helped start a company called Alnylam that is developing liver-based treatments using this very strategy. But if you want to treat cancers of the lung or somewhere else, that's tricky. Now, fortunately, in the Coke, we have many folks who are developing new strategies to deliver various things, including these small RNAs, to tumor cells outside of the liver. And Dan Anderson's lab has been one of the pioneers, along with Bob Langer's lab. And in Dan's lab, James Dahlman developed a particular nanoparticle that they showed was able to carry these small RNAs, not just to the liver, but to other places, including, importantly here, the lung. So Wen Zhu in my lab collaborated with James Dahlman and tested the ability of nanoparticles that were loaded up with microRNA 34 or siRNAs against KRAS, or the combination, to treat effectively the lung cancers in this model system. And sure enough, when we treat these animals with multiple doses, animals carrying established tumors, in contrast to control tumors that continue to grow, the miR34 treated tumors show growth inhibition. So this small RNA therapy actually works. It slows the growth of these tumors. Likewise, if we inhibit KRAS with this siRNA against KRAS, we can see a tumor growth inhibition. If we combine those two, we can actually see tumor regression taking place, an RNA-based treatment for cancer. This opens up a whole new strategy for the treatment of this and many other cancers. And with respect to longevity as well, we can expand the lifespan, as well as those drugs, in fact, better than those drugs that I showed you previously. Now, you would think that this would be an attractive strategy for pharmaceutical companies. We actually tried to attract pharmaceutical companies to support this work and translate this work. The scientists in that particular company were really interested, and then the lawyers got involved. And sadly, they killed the project for reasons that I can explain later. But this, I think, is a promising new approach for the treatment of this and hopefully many other types of cancer. So I want to switch gears and talk about this process of cancer genetics. Cancer genes, as I mentioned, are a very important process in tumor development most broadly. This subject has become much more complex of late, in part because cancer genome sequencing studies have revealed that many genes, I mentioned this before, many genes are mutated in various types of human cancer. In lung cancer, for example, the average tumor has about 175 protein-altering mutations. That's a bewildering collection of changes. And it makes it difficult for people like those in my lab to think about how to study that process systematically. How do we look at all of those different genes and their particular contributions to cancer? And the reason it's hard is that the way we do that, or have done that in the lab, is to make mice with mutations in those genes, and then to cross those mice with the mice that are cancer prone, and then test the contributions of those mutations to the tumorigenic process. That process would take 2 to 3 years for every gene that you want to study, and that was bewildering to us, and frankly a bit discouraging. And around this time, as we realized the daunting challenge that lay ahead of us, those in the lab became depressed. The skies above us were darkening, and we didn't know quite what to do. So I called Ted Cruz. And he said, "Look to the heavens and pray." So we did, and sure enough, the skies brightened, and delivered unto us was a tool, and it's called CRISPR. Now, I'll go through this quite quickly. CRISPR is a remarkable new tool available to biological scientists. It's based on a system in bacteria that leads to the inactivation of viruses that are trying to infect the bacterial cells. Various workers have perfected this system and morphed it into a form where it can be used effectively in mammalian cells. This work was carried out by many labs, including Feng Zhang's lab here at MIT, but important contributions by Jennifer Doudna and Emmanuel Charpentier, George Church, and others. We now have this tool which allows us to inactivate and actually modify by surgical precision essentially any gene of the genome in ways that we couldn't have envisioned before. And this technology is sweeping the field. There have been, since these publications, which were just about 3 years ago, 2,500 papers published on this CRISPR technology. We in the lab decided to see if we could make this work in our mouse models, and Thalis Papagiannakopoulos and Francisco Sanchez Rivera and colleagues decided to use the KP model to see whether we could make it work. And again, without going through the details of how it was done, we created a virus that carried the CRISPR components in order to bring those CRISPR components directly into the cancer cells as they were developing, and thereby hopefully inactivate any old gene of interest as those cancers were evolving. And they did that with a variety of different genes of interest, and that's shown here. And each time they did it, the cancers changed in a distinctive and measurable way as a function of the particular gene that we were targeting. In all, in this experiment, which took about 7 months to complete, after maybe a month of planning, we generated hundreds of cancer samples, which we could then interrogate by various biochemical methods and study with respect to their response to treatments. This same experiment using the old-fashioned techniques would have taken us years to do and cost multiple hundreds of thousands of dollars. So this has completely changed the way that we are prosecuting genes of interest in my lab and labs around the world. It is fast-tracking our understanding of cancer. One of the reasons we're excited about how quickly we will become able to come to solutions for cancer is based on this technology. I'm going to spend just a minute or so mentioning one particular collaboration that involves this phase of the process of cancer development, the metastatic phase, that last stage, that lethal phase of cancer. We're very interested in that phase, but I want to mention one particular project that I'm excited about because it underscores collaboration and technology. I was sitting around with colleagues in the lunch line at the Koch Institute, Scott Manalis and Matt Vanderheiden, explaining that it would be interesting if we could build a device that we could plug into the blood system of a mouse and see whether there were circulating tumor cells in the blood, to be able to measure them, quantify them, do it longitudinally to see what the change of those cell numbers were over time, see what their response to treatment was. This was sort of a pie-in-the-sky idea, but testament to the creativity of the Koch culture, Scott Manalis said, "I think we can do that." Sort of can-do attitude, and then he went off and did it. And that's what you're looking at here. This is a device which is plugged into the blood system of a cancer-bearing mouse that can actually measure circulating tumor cells in real time and longitudinally. It's not just an idea, it actually works. You can actually see these cells, and you can actually capture the cells by activating these various ports, and then you can study the biology of those cells using various molecular techniques, including, as we've done here in Alex Shalek's lab, looking at the RNA expression of individual circulating tumor cells. This is just an example of a technology that comes out of the kinds of collaborations that we have available to us in the Koch. I mentioned earlier that the interactions between cancer cells and other cells are very important. In particular, the immune cells. The immune cells of the body have the potential to recognize and eliminate cancers. The hope is that we can stimulate that to happen more effectively. And in fact, that is starting to happen. You might know that cancer immunotherapy is a very powerful new approach to the disease. Science magazine declared cancer immunotherapy one of the breakthroughs of that year. And the reason is that there are now new agents, including FDA-approved drugs, that can effectively stimulate an immune response against your cancer. Here's an example of one. This is a study carried out just this past year, which led to the approval of this particular drug, which is an antibody against an immune checkpoint called PD-1. And you can see patients treated with this drug do much, much better than those treated with standard chemotherapy. These patients with lung cancer have failed previous therapy. But when treated with this drug, even after a couple of years, 30% of them are alive, compared to the control where just 10% are. And we know from other studies that these patients are likely going to have durable cures, or durable responses, in some cases, cures. So these drugs have really started to change the way we think about our prospects for controlling cancer by stimulating the immune cells. The action is right here. There are T cells in your body that can attack foreign cells, and your cancer cells become foreign. If you can stimulate T cells to attack your cancer cells, that's a good thing. So we got excited about the prospect of studying the tumor-immune interface with our mouse models. And so Michael Dupage and Ann Chung and others built models to stimulate immune responses against cancer. John Proctor funded some of that early work. This was done by manipulating those viruses that we use to start the process at the beginning. And this actually does work. If you introduce into these tumors antigens that the immune system can see, you can attract immune cells to the tumors. You can actually get T cells into the tumor microenvironment. And they're not just bystanders, they're actually doing something. You can measure an anti-tumor response by those infiltrating T cells. They're attacking the tumor just like we want them to do for a while. But eventually, and not very long thereafter, they fail to sustain that response. The number of those tumor-reactive T cells drops. Their activity states drops. They become, and this is a technical term, exhausted. They're there, but they're not functioning. And so we decided we would investigate this process. And various investigators, Michael, Amy Lee, Nick Joshi, and others, have looked at this system in our lab. There are various ways that the immune system can get shut off in the development of cancer. Here's a list of them. These checkpoints get activated. Various cells of the microenvironment turn off immune function. One of those checkpoints that I actually mentioned, PD-1, does in fact get upregulated in the cancer cell, in the T cells at that late time point. But Amy Lee has actually tried that anti-PD-1 antibody, and it doesn't work in this system. So this is a challenge to understand why it doesn't work and what we might be able to do about it. Nick Joshi has asked whether a different cell, a different mechanism, in this case a cell called a regulatory T cell, might be involved in turning off the immune response in an established tumor. First, he measured the quantity of these regulatory T cells in the tumor and found that they were in fact highly increased in tumor-bearing mice at this late stage. There's about a 20-fold increase in T regulatory cells in the tumor microenvironment at the time the T cells get exhausted. That's guilt by association. But in these mouse models, we can do experiments. And Nick was able to deplete the T regulatory cells and ask what the consequences of getting rid of that inhibitory cell type was. This is the before picture. The tumor cells are nice and juicy red. The T cells in green are sitting on the sidelines, exhausted, not controlling the tumor. He could then get rid of the T regulatory cells by this method. And when he did so, he saw this. The immune cells now increased in number and infiltrated into the tumor. And when they did so, they caused massive tumor destruction. So T regulatory cells are controlling the immune response in this model system, likely in human cancers as well. And we're now trying to figure out ways of specifically inactivating regulatory T cells in the tumor microenvironment to be able to translate this for use in people one day. All right, I'm coming to the end of the hour. I just want to mention two very brief projects, both of which are nascent projects in the lab, a view towards the future, and also underscore the collaborations that we have available to us at the Koch. And the first involves a collaboration with Sangeeta Bhatia's lab on this process of early detection. If we could eliminate cancers at a very early stage, when they're, for example, surgically resectable, our success rates would go way up. But we need to be able to find those cancers earlier. And Sangeeta's lab is developing nanotechnology-based strategies to do that. And they've published some of this work, but not as it relates to the lung. So this is unpublished data. And what they do is to take an aerosolized nanoparticle and introduce it into, ultimately, humans, in these experiments, mice, into the lungs of tumor-bearing or non-animals. These nanoparticles have little tags on them, shown in red here, and these tags can be clipped off by an enzyme, this little Pac-Man thing, an enzyme that gets upregulated in the cancers specifically. If the tags get clipped off, they can now get excreted into the urine and detected in the urine. And then by a simple urine test, one can find the evidence of a tumor in the lung. Seems crazy, but actually, in a collaboration involving Andrew Warren and Tuomas Tamala, they tested this idea by allowing mice with these lung tumors to inhale the nanoparticles and then tested them either at 3 weeks when the tumors are tiny, or at 6 weeks, or at 7 weeks to see whether there was a signal of nanoparticle activation. And lo and behold, at 3 weeks there's not much of a signal, but at 6 weeks you can clearly see one, and at 7 weeks it's undeniable that you can detect cancers in these mice by this very simple nanoparticle-based aerosolized urine test. I should mention that we make the undergraduates collect the urine. Kidding about that. So I'm excited about early detection opportunities. And finally, I'm excited about prevention opportunities. I think among the projects in the lab, I'm excited about all of them. I'm probably most excited about the idea that we can actually prevent cancers one day. And this is a project that's in the hands of David Kanner in my lab in collaboration with Omar Khan and Jasdev Chahal, who work in the Anderson and Langer labs in the Koch Institute. And the idea, which I'm just going to give you in concept form, is that we've learned a lot about how these tumors evolve in this mouse model. And there's one particular protein shown here called HMGA2, which gets regularly upregulated in late-stage cancers. It's not there in the early-stage tumors, but as shown in the brown staining, it becomes expressed in more than half of these late-stage tumors. Now, the important thing you need to know is that HMGA2 is not normally expressed anywhere in the adult animal. It's only expressed in the embryo, but it becomes abnormally upregulated in the tumor. And so the question is, if we know that's coming, can we vaccinate the mouse such that when it sees that, it will eliminate those cancer cells? Prophylactic vaccination, preventative vaccination based on our knowledge of what's coming, just like vaccination against infectious disease. And so David and his collaborators are now doing the experiment of taking the KP mouse and vaccinating it with this protein, HMGA2, so we'll get an immune response against HMGA2. And then we would induce tumors in these mice and ask whether, in fact, we can protect them against the development of advanced cancers. If this works, it will suggest that we could do similar things in human beings. And one day, perhaps, we will. And if you like that idea, I encourage you to consider coming to our symposium in June on the subject of prevention and early detection of cancer. So I've come up to the end of the hour. I'm going to thank just 2 more slides. The work that I described to you requires tremendous resources, and I've received tremendous resources and support from many places, including, importantly, the Howard Hughes Medical Institute, the National Cancer Institute, the Lustgarten Foundation, tremendous support from the Koch Institute, including the Swanson Biotechnology Centers and other funders. And finally, I'm going to stop where I started. Nothing I presented to you could not have been done by the collective efforts of a tremendously talented group of people, and I want to thank them, thank you, and thank you again for the Killian Award.
Thank you very much, Tyler. I would ask people with questions. There are microphones in both aisles. There's one here and there's one somewhere right about where I'm pointing there. Please come to the microphones, Tyler will be happy to take some questions.
And if you prefer to shout, I'm happy to repeat your question.
This side.
Can you tell us whether or not cancers are at all found in embryos? And if not, is that significant, and does that tell one anything?
The question is, are cancers in embryos? Occasionally, yes, occasionally. But if you recall my earlier comments about the fact that cancers require the acquisition of alterations in genes, it's not surprising that you wouldn't find them very often, because the opportunity for an embryonic cell to acquire the necessary changes to convert it from normalcy to malignancy is very, very low. It typically requires a long time, years or even decades, to acquire the necessary damage. At some frequency, that will happen in short term, and so we do occasionally see it, including in embryos, sometimes in children, as you know, but those diseases are relatively rare.
If you're far away from a mic, just raise your hand. Yes.
So as you know, this immunotherapy, which is so exciting because it gives some cures, doesn't work in a number of cancers so far. Breast cancer, some hint in prostate, but in many others, there's no hint at all. Do you see the same sort of Treg effects in those tumors as you do in your model?
Yeah. So the question is a good one, Phil. It relates to our lack of understanding of why immunotherapy works when it works and not otherwise. So you're asking whether Tregs might contribute. I think the answer is likely yes. Certainly there are cancer epidemiological studies that would suggest that the presence of Tregs correlates with poorer prognosis, and that would suggest that Tregs may be inhibiting an otherwise effective immune response. I think that's part of the answer. There are likely other answers why in certain circumstances the immune system can see a tumor, in other cases it can't. The immune system relies on there being differences between tumor and normal. The more differences, the better. Lung cancer, where this treatment works, melanoma, where this treatment works, happen to be two of the tumors that are full of mutations because of the carcinogens of smoke or of the sun. So they're very different, and that might be why the immune system is particularly good at seeing them. But I think some of these other suppressive mechanisms are likely part of the answer as well.
Yes, I speak to you. Is this on? Is it working?
Yes.
Oh. Not as a biologist, but as an engineer and a recent pancreatic cancer survivor. Recently, the Vice President announced a moonshot approach. In reading the literature and the popular press, etc., I see a lot of negativity about the approach of this moonshot, namely that cancer is more than 200 separate diseases and largely unique to the individual. Can you give us a comment on how this is going to affect the future?
Yeah, so I think that, for one, we don't have a very clear understanding of what the moonshot effort will look like. We hope that it comes with a significant increase in funding, and that will allow a variety of things to be tried and tested and accelerated. So I think the details still have to come in to give you a proper answer to your question. I think some of the negativity relates to our general concern in the cancer research community of trying to put a timeline on anything. These are hard problems, and it's difficult to say, with more money, we'll solve the problem in 2 years or 3. Those statements have been made before, and I think they're unwise and not very helpful. So more funding and dedicated efforts in certain areas. Immunotherapy is on that list. Immunotherapy is very promising, but we have questions still to answer. With more funding and concerted effort to understand some of those questions, will you be in a better place to use those treatments, maybe even in the short term? Other things on that list are early detection, I mentioned, even prevention, I mentioned. Those areas don't get enough funding right now, and so maybe with some additional funds, we can shift the curve of diagnosis or even to prevention. So I'm not a skeptic, I'm an optimist, and I'm happy that the Vice President is talking about more funding. That's always a good thing.
Do I see further hands? Yes.
So, I guess one of the confounding problems with cancer, obviously, is that it's a disease of the self. And the immune system goes through a clonal selection process so that you don't have autoimmune disorders. So regarding this last vaccine trial effort, do you have any experience, for instance, indicating at which point do tumors acquire immune resistance or immune evasion? And is there potential, even with an earlier vaccine that's delivered, for tumors to form and maintain an evasion? Is it MGA2?
HMGA2. So what I showed you is a concept. It's in very early stages. You raised the question of self versus non-self and tolerance. We actually believe that we can create a vaccine against HMGA2. I won't talk about those data because they're very, very preliminary, but I think it's possible that we can at least get the immune system to see HMGA2. If we can, then the tumor will be in an ongoing discussion with the immune system. Sometimes the immune system will win, and I think that would be a good thing. Sometimes the tumor will win, and some of these immunoevasion mechanisms, Tregs and other things, will come into play. Maybe the tumors will lose the expression of HMGA2. But sometimes they will see a positive effect, and that might lead to a shift in the likelihood that somebody who's, for example, predisposed to a particular cancer will get it, or how long it will take them to get it. So these may not be panaceas. They may not solve the problem for the reasons that you're suggesting, but I think they might well help, and that's why we're pursuing it.
So I'll ask a question myself while I'm looking for further hands. So I see many in the room whom I take to be students and postdocs. So thinking of all the students and postdocs in the room, could you sort of look back on your own experience and talk about how you pick good problems or how they find you?
Well, what I tell my students and postdocs is a little bit about my comment about swimming in your lane and keeping your head down. I think it's good to keep your head up and swim out of your lane. I think it's good to go beyond, to challenge yourself and to push beyond the conventional. So my students and postdocs do things that people have never tried to do before. And sometimes it's hard and it takes a long time, but usually with a good idea and perseverance, you can get to the end. So challenging yourself to doing something difficult is part of the answer. Yeah, I think in addition, just related to the Koch concept, keeping your mind open to alternative approaches from other fields. I think I'm seeing this more and more, and I've given you some examples where if you just focus on what you know, you're blinding yourself to many new opportunities.
Thank you. Right in the middle there, and then I'll go back there. Yes.
Looking at the circulating tumor cells that you discussed and next-gen sequencing and ultrasensitive assays for DNA, do you think it'll be feasible in the near future to basically screen routine cancers using these methods?
Yeah, it's a great question. Circulating tumor cells, the answer is likely yes, even now. These cells are being used to track the state of an individual's tumor now. How many there are pre-post treatment, for example. Soon, the circulating DNA that you referenced will be used in a similar fashion. In fact, it already is being used in that way. And maybe before too long, circulating DNA will be used to test whether somebody's got an early staged cancer in the form of early detection. And one of our speakers at our symposium, actually, Bert Vogelstein, will talk about that very approach. So I think the answer is yes. You worry a little bit about what will you detect, because if you have an abnormal cell, it's not yet a cancer, do you really want to know that and intervene then? So we need a little bit more specificity about what the signal is telling us. But the answer is, I think, yes. Before long, these tests will be used, perhaps routinely. You know, your annual checkup could include sequencing of your circulating DNA. In fact, I'll bet on it.
And there was a question right there.
So my question is about the KRAS and the P53. We know they have many different types. So only contributor to the tumor genesis differently. Is there any quantitative data?
I'm sorry, can you say it again? I didn't hear the middle. It's RB and KRAS and P53, yes?
They have different, many different types of mutations.
Uh-huh.
And how they contribute to the tumor genesis differently.
Yeah.
Any quantitative.
Yeah. So there's a detailed question related to the specific mechanisms of activation or inactivation of KRAS and P53. Short answer to your question is yes, there are differences. So the exact way in which you activate KRAS changes its effect on the cell. So different tumors do it slightly differently. Two tumors of the same type that do it with slightly different mechanisms can have different responses. And P53 is similar, actually. The way that you inactivate it can influence the exact pathways that the cell will take.
Are there other questions? Right here, yes.
Yeah, there are many different kinds of mice that have been developed. Many different kinds of mice that have been developed in your laboratory, and if that's the case, that it seems to me that there are many different ways then that present themselves as to how to treat these mice and therefore develop a much greater likelihood that you can cure those kinds of mice.
Yeah. Well, that was one of the motivations, David, for creating these models. We needed to have better systems that we believed in, that were faithful to the human disease. And the answer is that now that we have them, the treatments that we can test, the likelihood that the results that we see in those models will play out eventually clinically is much, much greater. So we have much greater faith. And that's really important because right now, there are too many drugs to test in patients. Phil mentioned immunotherapy. There are so many candidates and combinations that could be tested, it outstrips the number of patients that could be tested with. And so we need better systems. And that was actually the motivation way back when, to create these models so we can create predictive responses for treatment to help guide us to figure out what to use eventually in patients.
Thank you very much. Let me ask everyone to join me in thanking you for a wonderful lecture. And congratulations again.